right. Welcome back to part 2 of our chapter 21. So we're going to talk about RNA and we'll talk about transcription. Transcription is the process of um, getting the genetic material out of DNA and preparing it to make proteins. So this is a ribosome. And the ribosome is the site of, uh, well, I guess the ribosome is the site of transcription. Wait, no, it's the site of translation, actually. It's where the actual protein gets made, but we'll talk about how that factors in. So this is probably a little after transcription, because then we get translation. And that's where we get tRNA and ribosomes together. But we'll get there, and we'll explain this. These are different subunits of the ribosome, and the ribosome is both protein and RNA. So I guess that's kind of why I'm showing it. Ribosomal RNA, or rRNA as we call it. There's a few different types, as you can see. tRNA, mRNA is going to be related to transcription, and ribosomal RNA, again, mixture of protein and RNA molecules inside these units, these subunits. Okay, so RNA. RNA makes up most of the nucleic acids actually found in the cell. So most of it is RNA, because to make DNA, you have to start with an RNA molecule. So there's already that reason. But also RNA serves a lot of functions in the cell, whereas DNA mainly just sits there in the nucleus being all condensed and stored up. So it transmits genetic information from DNA. So DNA isn't directly responsible. It operates through RNA. Several different types. We're going to go through them. And of course they are different from DNA. So the ribose sugar is a deoxyribose in DNA, and we know uracil is going to be here instead of thymine. What is new is that the RNA molecules are single-stranded. Where DNA had a complementary strand, we will see RNA molecules complementary pair with things, either other molecules or with themselves, but we don't see any double-stranded structures. And RNA molecules are much smaller than DNA molecules because, of course, DNA contain the entirety of your genetic code. They contain all of the instructions, whereas mRNA are sort of like if you had a book of instructions to how to run a whole store, and one of your employees needed help figuring out how to do one tiny little job, you opening the big book of instructions and then just photocopying the page they need and handing them that page. That page would be like mRNA, where DNA is the big book that you're keeping. And so mRNA is the way that the cell sort of sends instructions out everywhere. So they're much smaller. In my analogy, one page is much smaller than the whole book, right? So mRNA and tRNA and rRNA are just smaller pieces than DNA molecules. So I mentioned there were types. There are 3 major types of RNA. I think there are now like 8 types. There was like 6 when I was studying, and now there are microRNAs and small nuclear RNAs and lots of other little types of RNAs. But the main ones are messenger RNA. This represents about 5% of the total RNA floating around in a cell. And this is going to be the information to make proteins. We've got transfer RNA. 15% of it is transfer RNA. This is involved in making proteins. The transfer RNA's specific function is to bind an amino acid. So tRNAs are all responsible for having an amino acid that they bind to, and each amino acid has its own tRNA molecule. And then there are ribosomal RNAs, and these, probably because of their size and the fact that we need to be making lots of protein, contribute to the reason that they're the most prevalent. So again, we'll talk about what ribosomes do here in a minute. We'll start with tRNA, a typical tRNA molecule. So we can show it like a clover leaf, and I have a picture on the next slide to show you. It's kind of an L shape when you put it into a three-dimensional model, and I think I have both of those to show you. It has an acceptor stem, and I'll show you that too. So this is the clover leaf structure, two-dimensional. You can see that there is internal hydrogen bonding happening in different portions of this molecule. So what that means is there are parts of this RNA molecule that actually complementary base pair to other parts, forming these structures. You got to imagine how that evolved, right? How cool it was that this molecule evolved because of sequences. The different different nucleotide sequences eventually led to this structure. And the structure has a really cool job. So in 3 dimensions, it actually kind of twists around a little bit and has this sort of shape with 2 ends, an acceptor stem. This is the 3' end. I guess the 5' end's here too. But the 3' end is important because it has a specific sequence that is going to recognize an amino acid. And that amino acid will form a bond, an ester bond, with the 3' group here on the sugar. And so we'll basically get an amino acid attached here. And that's important because these transfer RNAs, tRNAs, their job is to bring an amino acid in and transfer it to a growing peptide chain. Now, the way that they know what order they need to transfer in has to do with the other side. And the other side of the molecule is what we call the anticodon. And it happens on this anticodon loop that's formed, right, from these hydrogen bond interactions. And it's a series of 3 bases, and so they're kind of shown here as just little nubs, but these are going to complement 3 bases on an mRNA molecule. So there's going to be an mRNA molecule, and every 3 bases will be recognized by a tRNA. And tRNA1, might bring in an alanine, and tRNA2 might bring in phenylalanine, and a tRNA3 might bring in another alanine again, depending on what this code says. And so the interactions between the mRNA and the tRNA happen here on these anticodons. Now, protein synthesis happens in 2 places, really. First, it depends on where you start the process of thinking about protein synthesis. Protein synthesis originates from the DNA. So in the nucleus, genetic information for protein synthesis gets copied from a gene in DNA. So here, let's say that here's your DNA, and you're interested in, digesting some glucose, and so you need to have some molecules that are going to help do that. And so some part of your DNA contains the genes for that machinery, for those molecules. And so transcription is the process that makes mRNA. So we're going to go over that process in a little bit. mRNA gets formed. Now mRNA, and depending on what kind of organism in us, we have processing to do. Some mRNA is ready to go, but ours requires a little bit of processing. We have to cut out some pieces, and we'll talk about that. And when those mRNAs are processed, they get moved out into the cytosol. So they get pushed out through the nucleus. In us, it would go through the endoplasmic reticulum. And the rough endoplasmic reticulum has ribosomes embedded on it. And that's good because ribosomes are where translation happens, where proteins actually form. But there's also free ribosomes floating around in the cytosol. And so these mRNA molecules are going to associate with a ribosome. We actually saw at the beginning of this, there's ribosomal subunits, and those will kind of clamp down on a mRNA. And then they'll move along the mRNA, reading its sequence, and make a big long protein chain, amino acids. That's what these little circles are. And it'll make a protein. And so, I guess that's the picture I'm showing down here. So translation is when mRNA actually gets turned into the protein involving the ribosomes. So that's the big picture. Okay. So and I think yeah. So this happens every time we want to again make a protein, to make a structure, mRNA. Now these mRNAs can get used over. There's often like several ribosomes at a time that are moving down these mRNAs. Like there could be a ribosome that's almost at the end that has its whole protein chain already, and then a ribosome right at the beginning that's barely starting. But eventually these mRNAs get broken down, like all RNA does. It's less stable of a molecule than DNA, which is why DNA was sort of evolutionarily chosen to be the long-term keeper of the code, and RNA to be more of a short-term. So these mRNAs get recycled often, and they can also be because you don't want your body constantly making a protein, if it doesn't need it. So these get recycled and broken down, and if you really wanted to keep making that protein, well then you keep making the mRNAs, and we'll talk about how mRNAs sort of get regulated here in a minute. So in transcription, similar to in replication, the DNA has to unwind. The DNA that has the gene in it is wrapped up in that double helix, and so we need to unwind it. And then the mRNA is going to get made from one of the DNA template strands. And I'll show you which strand here exactly in a second. We know that with mRNA, because it's RNA, it will have uracils every time there would have been a thymine. And then once this is formed and processed, it moves out into the cytosol where proteins are going to be formed. So if we look here at the DNA strand, and we look at the sort of replication fork, we don't want to really call it that, I suppose. Transcription bubble is the new term, because we're not replicating. You'll see that there's an informational strand and a template strand. The copy gets made from the template strand, okay? Because we want the mRNA to have the same info that the informational strand has. So if and I guess I'll show you it with a real example here in a second, but let me see if I can sort of figure this out. If this was the template strand, if this is the template strand, then the informational strand would have had a T, and it would have had an A, and it would have had a C, and a G, and an A, and a T, and so on. Right? I'm just pairing with these guys. So it makes sense that our mRNA should have the same information. UAC, TAC. Remember, U is substitutor for T. GAT, this would be GAU. And so the same information that would have been on the informational strand. So the template strand is the one that's copied. It's also important to note here that the informational strand is in the 5 to 3' direction. And remember, this is the direction that we write, that we make mRNA, or make the phosphodiester bond. So the only way for that to work is if we read along the 3 to 5 strand, so that we can write the 5 to 3 strand. If we were writing along the 5 to 3 strand, instead, we would have to be synthesizing that other template, in reverse, and we don't have enzymes that do that. This RNA polymerase isn't able to do that. You can see here it writes in a 5 to 3' direction, just like DNA polymerase. So in our bodies, we have exons and introns that are part of our DNA, in our genes. And the exons code for proteins, the introns are just sort of space-filling, we don't really know exactly what they do, sort of sections of our DNA. Here, I'll show you. So for us, the pre-mRNA has all of these pieces sort of copied in, and the processing cuts out the introns. So all the non-coding parts, the introns, they get removed, and then it gets sort of pasted back together, and then that is our what we'll call mature mRNA. And this goes on to the ribosome to make a protein. Regulation of mRNA, so mRNA synthesis, right? It's actually controlled what we call at the transcription level. So there's transcription and translation, right? Transcription was making mRNA. Translation is reading the mRNA. So it's at the transcription level that we regulate protein synthesis. And basically, there are specific mRNAs that get synthesized when a cell wants a particular protein. So the transcription of a gene requires RNA polymerase to bind to DNA in both plants and in animals, and to know which gene it's reading, and to, I guess, know what protein it ultimately wants to make. So there are a group of protein complexes that we call transcription factors. And these transcription factors, they have to recognize and bind to a region of the DNA, or up kind of ahead of the gene, called a promoter region. So every gene would have its own unique promoter region. There would be transcription factors that help to call and recruit RNA to that site. That would be that would recognize that promoter region, and those transcription factors would be controlled similar to how we would control enzymes. There would be allosteric sites on those where maybe if this makes a protein, that protein, or if so these would be like activators. This little thing here, this might be an activator that turns this transcription factor on, so it'll go bind to this promoter region, or that activator could potentially change the shape so that it doesn't bind anymore. And by modifying your transcription factors and their ability to land on the promoter region and to copy that gene, you can then sort of affect the rate of how much mRNA you make. So without getting too into it, it is a complex. There are more sort of little regions here, and there are a lot of transcription factors that are involved. Sometimes there's upwards of 10, 12 different transcription factors that all have to be in a certain spot so that a gene will get copied. But that's the whole point, is you want to maintain control over which genes and whatnot. And think about the similar feedback mechanisms for enzymes. Like, late products end up being inhibitors of the cycle, right? So that's true here too. For example, if you are an organism that can that eats glucose, but suddenly you're in a place where there is no glucose and maybe there's lactose, the lack of glucose will probably start to affect a transcription factor that would normally be turned off in the presence of glucose, and now it won't be turned off anymore, and its job might actually be to go and start making molecules, basically the machinery, to start breaking down other sugars, because if glucose is around, all of that machinery would get turned off because we don't need it. But now that glucose is gone, suddenly transcription factors that code for, or that recognize genes that would maybe make an enzyme for lactose, or like a lactase enzyme, start getting turned on, and that sort of thing. So the products of the translation sometimes can be inhibitors and promoters even of these transcription factors, and so it's all very kind of closely regulated. Okay, what is the sequence of bases in mRNA that would be produced from this template strand of DNA? So this shouldn't be too difficult. You're looking for the one that you would basically you would want to write a 5' to 3' mRNA strand here. So pause it, give that one a shot. Shouldn't be too difficult. Hopefully you came up with number 2 there, realizing that mRNA has uracil and the thymine wouldn't be included. This one just doesn't match the sequence, right? Or doesn't match as a complementary. Good. So let's talk about the genetic code. So I mentioned the tRNA's job here. You can see a little bit better the tRNA here. Actually, this would be a serine tRNA, and so this one would hold a serine amino acid. And again, the function of these is to facilitate the task of synthesizing proteins. So we have the mRNA that brings the message from the DNA. We have the tRNAs that bring the amino acids, and we have the rRNAs, the ribosomal RNAs, that we're going to look at here in a minute. All right, activated tRNA is what we call it when it has its amino acid on it. It's deactivated or unactivated when it does not have that amino acid bound to it. So let's go through this step by step. So inside the nucleus, pre-mRNA is made according to the transcription and all that stuff. The template strand, promoter regions, so we get the mRNA. It gets processed, and then it gets, so the introns are cut out, and then it leaves the nucleus. So now it's floating around in the cytosol. So it's going to attach to a ribosome. So the mRNA is going to attach to a ribosome, and I think I have a picture of the ribosome attaching here in a minute. After it attaches to the ribosome, this is where protein translation is going to happen. We have the activated amino acids start coming in. Now, here is where we need to look at the genetic code first. So what are these anticodons actually recognizing, and how does that turn into protein? How do we know what the protein sequence will be from looking at the DNA, right? So if we look at the genetic code, what we're going to see is that it's basically 3 nucleotides. We call them triplets. Three nucleotide or triplets are what we're going to call codons. So I mentioned that the anticodon is what is on the tRNA. The codon itself is going to be on the mRNA. So there are, you've got your mRNA, and you've got your sequence, right? And these are A's and U's and T's and A's and U's and G's and C's, and every triplet is a codon. These are codons. Now, there are 20 different codons for all 20 amino acids, and the genetic code actually even has stop signals. So, like UGA or UAA or UAG, if this happened to be the codon that we get to, or as we're reading the sequence, this would signal the end of the protein synthesis. There's a start codon. It's always the same, AUG. That signals the start of protein synthesis. And realize that mRNA actually has a little bit in the front and a little bit at the end, both to protect the integrity of the code while it's floating around, but also to give something to latch onto when you're going to start reading these codes. So, the ribosome is sort of going to scan across this mRNA until it finds that start codon, and that will signal the start of the protein synthesis. Now, to determine the amino acid sequence, you would look at the section of mRNA, you would spread it out into the triplets, and then you would look up on this table what that would equal. So like CCU, when the first letter is C, the second letter is C, and you go to this little square here where they interact or intersect, and you can see what the third letter can be. Actually, when it's CC anything, it's always proline. So the first one here would be proline, right? AGC, so A and G, so in here. AGU and AGC is serine, AGA and AGG is arginine. It's interesting that when they get grouped, the third codon here, for the different groupings, you'll notice that it's a purine or a pyrimidine for those different groupings. The purines will get one, pyrimidines will get another. I find that interesting. Some of these amino acids have 4 different codons that code for them. Some amino acids have only 2. Is there one that has 6? No. There are some stop codons spread throughout here. There is a start codon, that's methionine. Yeah, I guess that's it. So this sequence would be proline, serine, glycine, leucine. And again, you can go through each of those letters and figure out which code it would be. I guess you could look at this column too. So that's how we sort of distinguish the sequence. Now, again, molecules of tRNA that have anticodons, they picked up the specific amino acid that they bind to, and then when their anticodon is recognized by the mRNA sequence, they attach to that mRNA. And I do have a diagram here of the ribosome. So here we kind of have to use our imagination a little bit because this sequence is already grown pretty long. So we missed the start sequence, but there would have been an AUG. I think that's what I said it was, right? AUG for start. So the first amino acid would have been a methionine, so we can kind of pretend that this is the very first one. So if this was AUG, I guess red's not the best color here. If that was AUG, then this would just be the anticodon for methionine. This would be methionine. And all of this is happening on the ribosome. The ribosome sort of comes in with its multiple pieces, clamps down on the mRNA, and starts to move along the mRNA. Now, the ribosome has 3 sites. It has the exit site, it has, I think we call the A site and the P site. Or maybe it's the peptidyl and apeptidyl site. Actually, I don't remember what the initials are. But there's 3 sites. There's the site where now a tRNA no longer has an amino acid on it, and so it can float away. There's the site where the next amino acid or tRNA comes in. What happens is the tRNA that comes in with the next amino acid, it gets everything transferred onto it. So now this little guy here will have the whole chain attached to it. This one won't anymore. And when the ribosome slides over by 3, this guy will leave. So the sites are just on the ribosome, and as it slides across, there's always a spot for the next tRNA to come in. There's always a spot for the tRNA that's sort of right in the middle where we're going to transfer the amino acids over. We're going to form a bond, right? A peptide bond between those. And then there's the exit site. And so it'll roll along this mRNA until it gets to a stop signal. And if it gets a stop signal, then there's no tRNAs that recognize that. So at that point, there are a couple release factors that come in, and then they disengage. The whole thing kind of falls apart. The protein floats away. A ribosome goes to find another mRNA. All right. And then, of course, the tRNA gets recycled. The tRNA with now nothing on it goes back into the cytosol, finds another free-floating amino acid, binds to it. Now it's ready to go and add to another protein. So this table sort of summarizes the nucleotide and amino acid sequences in protein synthesis. So you can see just how the informational strand differs from the template strand, right? If you were copying the template strand, the codons wouldn't match up. And if you read, or if you copied the informational strand and wrote like a complement to that, you would see that the amino acids don't match up either. So it's important that the mRNA has the same info as the informational strand. So this is why when you're copying DNA to make mRNA, it's crucial you're reading the 3' to 5' direction. If you're reading the 5' to 3' direction, you're automatically getting a bunch of gibberish that isn't going to match to those amino acids. I mean, you'll find matches, but you won't get the right protein sequence. Notice that the tRNA anticodon basically matches the template strand. Okay, quick learning check. Write the amino acids that are coded by this section of mRNA. So I think I don't take into account here what I literally just told you guys. Oh no, this is mRNA, so you're all good. Be aware that on the test, I love to give this question, but it's a section of DNA, which means your job is to first write the inverse of this so that you can turn that into an mRNA. Unless you guys are comfortable just taking this and turning it into an mRNA, realizing that anywhere you see a T, you would just turn that into a U. But be aware that when you're copying DNA, that's what you're looking for. You want to make sure that the sequence is right. Also, don't assume that the first three letters are going to be the first three that you turn into an amino acid. On the test, I like to give a big long DNA strand, again, DNA, and you have to scan across to find a start codon. That start codon might be here. And then every three is your genetic code, right? Not just the first three, first three, first three. So that's your warning. For the exam, I give you DNA, and you have to find a start codon. You're looking for AUG to start your sequence. But the answer here, if you did it, since this is mRNA already, this should just be alanine, valine, asparagine. All right, last but not least, let's talk about, well, I guess it's not last. Let's talk about mutations, and then we'll talk about viruses and illnesses. So genetic mutations. This peacock has albinism. It doesn't produce melanin, so it doesn't make bright colors for its feathers. And the reason it doesn't produce melanin is because it has genetic mutation. So an error in its DNA that leads to faulty proteins. So a mutation is a change in the nucleotide sequence. And this can happen because when it's being copied or being transcribed, an error happens, or this can happen from things that cause mutations, like mutagens. Radiation can cause mutations. Chemicals can cause mutations, and viruses as well. So again, mutations can cause changes to sequences, which is going to lead to changes in proteins. Since enzymes are made of proteins, enzymes can also be affected and altered so they no longer work. The new cells of the person may not survive. This can either lead to disorders or diseases, or even death. Usually if it's death, it happens in development pretty quickly. Okay, let's look at some normal DNA so we can see how different mutations and different types affect the sequence. So normal DNA, transcription would yield an mRNA, translation would yield some protein sequence. Nothing really new to say there. Now there are two types of mutations that can, well, the first two types that we'll talk about are point mutation and silent mutation. So pay attention to this glycine here. We're going to have a problem with glycine here in a second. So a point mutation is what happens when one amino acid, one nucleotide gets substituted for another. So in the previous slide there was a cytosine here, but because of a mutation, a thymine is there instead. So we get a mutation. So instead of then putting in a G here to complement the fact that there was a cytosine, now we're going to put an A. That A happened to be the beginning of a codon, and if you remember back to the genetic code, it was the first two letters that sort of determined what column or what box we were in, and then the third letter determined like if it would be a different amino acid or just the same amino acid. In some cases, or in a lot of cases, the third codon didn't even matter, right? The third letter, we kind of call that the wobble or the wiggle room. It's a way that the genetic code sort of preserved itself and kind of maintained. I guess it was like a not proofreading. I guess it's just a mechanism for minimizing errors to have sort of redundancy. But anyways, this isn't that case. This isn't the third spot. This is the first spot, and that changes everything. It changes a glycine into a serine. Now, if this happened to be in an active site of an amino of a protein, a serine has an OH where a glycine doesn't. A serine is going to form hydrogen bonds where a glycine wouldn't. And so this is going to be a problem. This is going to inactivate or cause some kind of misfolding. And so this is a point mutation. Now, there are also silent mutations. Silent mutations can happen when they don't change the amino acid. So for example, rather than this happening the way it did, what if it was the G that got switched into an A? That would lead to us putting a T here instead of a C. And if I take a look at the genetic code real quick, what is AGT? AGT is, oh, it would be AGU. My bad. It would be AGU because there is no T. AGU would also be serine. So I suppose, oh, but this is if, so now I'm just looking at, if it was serine before and then a mutation, what is it now? It'd be serine still. I forgot that this used to be glycine. But, so there are some mutations, again, substituting a different letter here, maybe in the wobble spot, the third codon, that actually lead to the same amino acid. So that's a mutation nonetheless, but it's silent because it didn't have an effect. The same amino acid was put into the protein, so the protein is identical. You can imagine mutations adding up over time, right? And eventually leading to changes, but silent mutations probably happen all the time and they go unnoticed. In addition to single mutations or point mutations and silent mutations, which are a type of point mutation, there are deletions and insertions as well. A deletion is a base being deleted from the normal code, and an insertion is a base being inserted into the normal code. What happens when we take something out or put something in is that all of the amino acids after that point, nucleotides, after that point, get shifted over. Okay, if we normally had, so I guess for comparison purposes, this was an AAA, so that led to a UUU, and that was phenylalanine. But if one of those A's is now taken out, we see AAC instead, right? And now CGA will be the new code that we will read instead of CCG, which is what it used to be. And instead of ATT, we're going to see TT whatever else. And so it shifts everything. We actually call these frame shift mutations, deletions and insertions. Insertions for the same reason. If I add in something here, it not only changes that three-letter code, but everything after it now is shifted over by one. These typically lead to proteins that don't work anymore. There's not a lot of scenarios where a deletion or an insertion doesn't have a giant effect. Okay, maybe if this happens at the very end of a protein, it might not be a big deal, but anywhere in the middle, this is sort of a death sentence. Okay, some of the effects of mutations. So some mutations don't cause significant changes in the primary structure of a protein. So in that sense, the protein might still function. But if they do cause drastic changes in the amino acid sequence, the structure might lose its biological activity, might lose its active site. If it was an enzyme, it may no longer function. It may no longer catalyze reactions. Substances may even form new shapes and actually pack together or aggregate. They can accumulate into the cell until they're poisonous because they don't break down or the thing that the enzyme was breaking up or breaking down might pile up and cause toxicity. So that can lead to, you know, um, a lot of kind of effects. And again, your DNA from X-ray, from UV, from mutagens, from viruses can all down the road lead to, defective enzymes or proteins, which can lead to, again, some cancers or some diseases. Let's talk about some diseases. These result from defective enzymes caused by mutations in the genetic code. So if the enzyme, for example, that converts tyrosine to melanin, like we were talking about for the albino peacock, if your tyrosine to melanin enzyme is defective, then no melanin gets produced. You would need tyrosine for the formation of melanin, but if you, as another example, if you don't have phenylalanine hydroxylase, then your phenylalanine doesn't make tyrosine. So if you don't have tyrosine, then you can't make melanin. So this can happen anywhere. I imagine there would be another enzyme here that might be affected. If the enzyme that converts phenylalanine into phenylpyruvate is missing, then you could potentially build up some other kind of, your phenylalanine might build up to toxic levels, or you might have a deficiency in phenylpyruvate and not be able to digest something. Every one of these things can throw an imbalance. Just a list here that if you're interested, you can look at some of the genetic diseases, right? These are all caused by just changes to DNA. Some of these are really crazy, like Down syndrome, right? This is changes. I think actually this results from having 3 chromosomes instead of 2. You're supposed to have a pair of each of your chromosomes, but if you end up with an extra copy of one of those, that leads to something like Down syndrome. Sickle cell anemia, I don't know if it's on here. Sickle cell anemia, I think it's just one amino acid gets changed in the protein for hemoglobin, and all of a sudden it's reduced oxygen carrying ability. It aggregates and breaks, it plugs and breaks blood vessels. But again, all of these are mutations of DNA. Learning check. Identify each of the following as either a point mutation, an insertion, or a deletion. So cytosine entering the DNA sequence. One adenosine being removed from the sequence. Or a base TGA changing to TAA. So pause it, give it a shot, shouldn't be too hard. Insertion, deletion, point mutation. So let's talk about recombinant DNA. So there's a few things we can do with DNA. One is we can cut out genes that we like and mix them with the DNA of other organisms so that they can have new powers, right? New genes that they didn't make before. There's reasons that we do this, and I'll show you a few. Viruses essentially can do this too. They are like recombinant, the method that we use to introduce DNA, foreign DNA, into an organism is similar to what viruses do with their own DNA. They insert it into our DNA. But we'll take a look at viruses separately. So we can recombinate DNA. The other thing that we can do is we can use DNA kind of like a fingerprint, right? We can look at your exact DNA sort of profile, and we can match it to, if we had some of your DNA, we could do all the same stuff to it, and then take a look at the 2 pictures and see if they match it. And by stuff to it, what I mean is we first need to amplify whatever part of the DNA we want to look at. For example, we like to look at the DNA that codes for our ribosomes, because for the most part, if your ribosomal DNA got mutated or changed, there's a good chance your ribosome wouldn't work anymore, and you probably would have died, because you need your ribosomes. So those largely remain what we say conserved. They don't change very much over time, because changes usually lead to changes in the activity, and since this is one of the most important enzymes that we can possibly have, or important molecules, ribosomes, their DNA tends to be pretty constant throughout history. And in fact, it does change a little bit, and that's how we're able to kind of do lineages and stuff and see where people have moved and where they come from and how they're related. But if we take a look at your ribosomal DNA and we take a look at mine, they should be different from each other. If we take a look at your ribosomal DNA from a cell that you have, and then we take a look at one from a sample that we have, and as long as we're looking at the same, like if I find your hair on the floor or your cheek cells, I can isolate your ribosomal DNA from that, just like I can take a sample from you directly and isolate your ribosomal DNA. We need to first start off looking at the same thing. Then we need to amplify it big enough so that we can actually do stuff with it and look at it like this guy is in this picture. What he's looking at is actually a chromatography. They put a little smidgen of DNA in these little cells, and then they run a charge through them, and they slowly start to migrate. And the reason that there's all these little bars is because they add something called restriction enzymes that cut them in all the same pieces. If I have two sections of DNA that are both from me, and I put the same restriction enzymes in them, they're going to get cut into the exact same pieces because they're both me and they're both the same gene. They're both, my ribosomal RNA gene. And so when we run them through a filter that has different sizes, the little pieces will move far, the fat pieces won't move as far, and we end up with this kind of banding that you see here. If it's both my DNA and it's both the same gene, they'll band exactly the same. We can use that fingerprint-wise to make matches of DNA. If there was two people and your DNA had a difference than mine, right? But we still applied all the same restriction enzymes, it would cut yours into slightly different pieces than it would cut mine, and the banding would be different. So we can use DNA to tell if two samples are the same, so long as we put them through the same process. We cut them in the same, using the same methods, and again, all the differences will show. If it was the same piece of DNA, then all of the cuts will happen at the same places because it's chemically driven, just like we have enzymes that'll recognize certain places on a protein to cut the peptide bond. There are certain what we call restriction enzymes that always look for certain sequences and then cut at those sequences. And so if those sequences happen in different places for you and me, then the sizes of our little DNA fragments will also be different, and this fingerprinting will be different. So let's talk about those processes. So for recombinant DNA, this is taking DNA from one organism and combining it with another. Restriction enzymes are used to do two things: to cleave the gene that we want. So here's the gene from the organism. Let's say that it's right here. We want this gene so we can stick it into this organism's DNA over here. So we're gonna, and actually we use plasmids, so maybe I'm getting ahead of myself. Let's do it this way. We've got circular DNA for bacteria. E. coli has circular DNA. They also have little plasmids, which are like little packages of DNA that they can share with each other, and they often put little I'll give you an example. I'll give you a real example. Let's say that I want to include a new gene in this E. coli. So E. coli up here has its DNA. It's a big circle mess. But I want to give it a new program. I want to give it a new function. Let's say that it dies if I put it in penicillin. So what I've done is I've taken an organism that has a gene that actually codes for a protein that breaks down penicillin. That breaks penicillin. That means any organism that has this protein, if it was in penicillin, it would just break down the penicillin and the penicillin wouldn't hurt it. E. coli doesn't have that. But I can give it to E. coli by using a restriction enzyme that'll recognize the sequence of the beginning of the gene and cut it, and recognize the sequence of the end of the gene and cut it, so that I get a little piece of a gene, or a little piece of the DNA that has the gene on it. that same restriction enzyme, I would use it to cut a place on the plasmid. So the E. coli have these plasmids that they can share with each other. We would use the restriction enzyme to cut a little spot out that I could go and stick my new program in, my new protein. So now I have this plasmid that has a protein from a different organism stuck on it. And we would just put this back into a mix of E. coli, and an E. coli would pull this into their cell. They would incorporate this into their DNA. They would divide, and now there would be two E. coli that had this. They would share this plasmid with each other through a kind of form of sex in a way. The little tube comes out from one cell to the next, and through that tube, these plasmids can be transferred to each other. In effect, like I could have a jar of E. coli, and I can put one E. coli in here that has this program, and by the next day, all of them will have this program. They can share that with each other. And so I've effectively turned these E. coli into something that can now survive in antibiotics. And so here's a little better picture. We isolate plasmids, we cut out little pieces of them, we find the DNA that we're interested in. It's like a program we want this new bacteria to run, right? We have bacteria running insulin programs, making insulin so that we can harvest it. We have bacteria that we have making enzymes that break down plastics. We have bacteria that have enzymes in them making, what else do they make? They make medicines, actually I have a slide that I'm going to show you. But we basically like hijack their cells and their bodies to make products for us. It's kind of messed up, but that's what recombinant DNA can be used for. It can be used for a lot of things too. I mean, you think about it. You could find, if we could treat a plasmid like our body, like our DNA, and find a way to go and cut out a faulty gene that makes, like, let's say a sickle cell hemoglobin, and then insert a healthy gene, ideally that would mean that you could cure sickle cell. The problem is, how do you get this into every single cell of the body? You know, we're still working on those kind of things. It's easy with a bacteria where they have one cell. You just stick this in, and now the whole bacteria is changed. But that's what recombinant DNA is used for. Now you can see here a few different areas of that. We make insulin, we make influenza vaccine, we make interferon, human growth hormone, human clotting factors, epidermal growth factor. All of these are things we make inside of bacteria. We used to have to smash up like dog and pig, gallbladders or livers or pancreases or something to get insulin. So like insulin from the old days was harvested from animals. We just make it now in bacteria. Okay, so that's cutting it up and putting it inside another organism. In order to get enough of the gene to stick into an organism or to make a fingerprint sort of picture, we need to make lots of copies of the DNA or lots of copies of the gene that we're interested in. And polymerase chain reaction is for that. So PCR, as it's abbreviated, is a process where in a short time we make multiple copies of a DNA. So it involves heating the DNA to separate the two strands, and then in the first cycle we lower the temperature and we add a bunch of nucleotides, so like DATP and DTTP and all of the ingredients that we would need, and we just let new strands form. And then we heat it again and those split up, and then we just keep adding ingredients and we let new strands form. And so it doubles and then doubles and then doubles and then doubles the DNA. And so this allows for, first, like if you have a big long section of DNA, like I was mentioning, if we wanted to compare, your cells to my cells, first we would go and just cut out, we have restriction enzymes that would recognize, let's say, the beginning of our ribosomal genes. So we would just cut out a ribosomal gene, or we would just, try to anyways. And then to that, we would add primers. Primers are little snippets of starting RNA that would stick on to the part of the gene that we just got rid of. So PCR has to have a primer also that's not kind of shown here. But the primer is going to go and find the part of the DNA that we want to copy, and then when we do the heating and cooling, it'll actually copy that gene. And then the primer falls off, and then the next time we do the splitting, the primer will go and land on these little spots again, and then we make more copies and more copies and more copies. And eventually, what started out as just having an entire section of your DNA ends with just having millions and millions of little copies of just the gene. So that's the bonus. We do end up with some full-length copies in here, but because of the way the primers work and because of all the cycles, eventually we just end up with nothing but gene. So then, this can get us a large amount of DNA. And we want a large amount of DNA so that we can then stick them in the machines, and they're called gels. We put them in a gel, and then we put a charge through it, and then it bands into different, sort of areas, and then we can find, if we have references, we can say, oh, something showed up in this area, that might be this gene, or we can say, oh, your pattern looks just like this other pattern, this must be a match. There's a lot of different things that we can do with this. But by being able to do this and screening for genes, present or not present, we can associate that with sicknesses. And they've been able to do that. Like there are two known breast cancer genes, BRCA1 and BRCA2, that if you have defects in those genes, you're at high risk for breast cancer. So DNA fingerprinting is like really close to what we've been talking about. So restriction enzymes, we build up a big sample using PCR, and then we place them on the gel, and then the banding pattern is unique to that individual. And so that's how we can do, and I think here they probably have like 8 samples all next to each other, and one of them is probably a control just to make sure that it worked. Like if this column here didn't go all the way down to the bottom, we might have to say, oh, well, none of these are accurate. So that's kind of how these usually work. There's a control, and there's like 3 or 4 samples next to each other. But you could see everywhere where they're the same. You could see everywhere where they're different. You can see, you can imagine. You could use it to do some comparisons. All right, let's talk about viruses. Viruses, like this one, can cause cancer. This is actually this one was caused by or invented by I was going to say Jeffrey Epstein and Roseanne Barr, but you probably don't know who those people are. Just kidding, anyways. Herpes virus 4. This one can cause cancer. We'll talk about how cancer is caused as a result of viruses and mutations here in a minute, but let's talk about viruses first. So viruses are small particles of RNA and DNA that contain about 200 genes, maybe maximum, and they can't replicate without a host cell. So they're missing all of the machinery to actually do stuff with their genes. They're just like floating instructions. And a lot of these are protected. These viruses have measures of surviving, and I don't mean like eating and stuff. They just have protective proteins that keep them safe on surfaces and whatnot until they can get into a nice environment where then they attack. So viruses, they need a host cell to replicate. Infection is just what we call it when you have viruses replicating inside you. They can replicate in cells, and then they basically take over the machinery and the materials that you're using to make your proteins, and then they make their own proteins from it. Viruses can either incorporate their DNA into your DNA and permanently be part of your cell now, or they can just basically kill your cell by busting open the cell and letting all their virus parts out. So there are a couple of kinds of end results here. So you can see here the virus, through various methods, can attach onto your cells and then inject its DNA inside you. Once the DNA gets inside, your body just treats it like every other DNA, and it transcribes it, it translates it. So using your ribosomes and your DNA polymerases, it makes new virus particles that then, again, bust out and go and infect other cells. So yeah, that's what viruses do. Now, the differences in how they enter into the cell depends on what kind of virus it is. Some of them have like literally like syringe type apparatuses that will inject a barb into the cell and then squirt the DNA in. Others can actually bind and then be endocytosed, meaning that the cell actually brings them inside. Once they're inside, their nucleic acids will mix with ours, and ours can't tell the difference. Protease processing proteins produce a new protein coat, so some of the instructions that virus carries, of course, are just how to make more virus parts. And the new virus parts get released into the cells. Now here are some diseases caused by viral infections. So chickenpox is a virus, common cold virus, hepatitis and herpes, HPV. AIDS is a virus. We'll talk about retroviruses here in a second. So a lot of different things can be caused by viruses. Now, viruses don't just infect humans, they infect bacteria as well. In a lot of cases, bacteria that aren't infected with a virus aren't harmful, and then sometimes they get infected by a virus, and that's when that bacteria becomes pathogenic to us. And so it's a pretty neat dynamic. There's a lot of protection that microorganisms have developed in response to viruses, and it's an ever-going sort of evolution driving viruses to be more and more creative. But yeah, they infect and make other organisms sick, not just us. Reverse transcription is a neat thing that viruses can do. So a retrovirus is a virus in which they don't have any DNA in their cell. They don't use DNA as their main information storing molecule. They use RNA. And the problem with that is that RNA that stores information can't be transcribed, right? We don't have something to look at RNA and make mRNA from it, right? So a viral RNA, a retrovirus has to have its viral RNA first turned into DNA, and it uses something called reverse transcriptase to do that. The reverse transcriptase makes the DNA strand from the virus RNA strand, and then that DNA strand can form a complementary DNA strand so it looks like regular DNA, and then our body will use its DNA polymerase and transcription factors and RNA polymerase to go and turn that into mRNA and make proteins out of it, of course, virus proteins. The HIV virus is a retrovirus. This one destroys and infects our T4 lymphocyte cells. This is why it's so bad, is because it wipes out cells that would normally be helping us fight infection. And so once you're infected with this virus and you have a decreased number of lymphocytes, you're susceptible to other sicknesses. This virus comes with its own reverse transcriptase packaged along with it, so that when it gets into the cell, the RNA can already be converted into DNA. Now this one you can see has an envelope protein. Some of them are encapsulated, some viruses are not. This lipid membrane probably helps it interact with the outside of our cells that also have lipid membranes. All right, but anyways, this is a diagram of a virus here. They do also have some pretty neat geometric shapes if you ever Google them. So after a retrovirus infects or injects its viral RNA into the cell, it forms the DNA strand, and then that turns into double-stranded, and then it gets copied. So when the cell replicates, the provirus, which is what we're calling it now, the double-stranded dRNA, that provirus produces more viral RNA to produce virus particles. And so you can kind of see not just the steps in green, but also some of the inhibitory steps that we can take. So virus enters viral RNA, reverse transcriptase turns it into viral DNA. Now it's a provirus. That provirus incorporates itself into our complement of DNA, and then it gets copied. And so when we copy things, we make just normal proteins, but we also make the viral proteins. All right. Um, what next? Treatment for AIDS. We can talk about treatment for AIDS specifically. This is based on attacking the HIV at different points in its life cycle, either inhibiting its entry, inhibiting its ability to turn its viral DNA into RNA into DNA, inhibiting its ability to package its proteins. Um, so that's one way. So just using different inhibitors at various stages of the life cycle. And then, of course, we developed some nucleoside analogs. I'm going to show you a picture of those. So the nucleoside analogs have mimicking structures. So they kind of look like the nucleosides that we would use for DNA synthesis. So drugs based on nucleoside analogs include AZT, which is similar to thymidine, DDL, similar to guanosine, DDC, D4T. I think I'm showing you these here. So all of these, what they do is they get incorporated into the growing polypeptide or polynucleotide chain, right? The virus RNA is being turned into virus DNA. And if these guys are floating around and they get stuck in to that growing chain, notice that they don't have 3' hydroxyls. Without a 3' hydroxyl, you can't add anything else to this chain. It stops. And so this effectively stops the formation of the viral DNA. So it stops reverse transcriptase. Reverse transcriptase sees these and can't do anything with it. So it's one method that we have. And so basically, when used in combination with like entry inhibitors and reverse transcriptase inhibitors and protease inhibitors, these analogs also effectively help to keep people who have that infection, the HIV, relatively healthy or symptom free. At least keep the virus from getting a bigger handle on their system, So cancer is when cells begin to grow and multiply without control. They can form tumors. They can spread to nearby cells. When they aren't spreading or being overly aggressive in their size and that sort of thing, they're called benign. When they are invading, they're called cancerous or malignant. Cancers can be caused by either chemical or environmental substances. They can be caused by radiation sources, so X-rays or ultraviolet UV from the sun, or from tanning beds, that kind of thing, or from viruses that are oncogenic, meaning cause cancer. Here's a couple of carcinogens. These are chemical compounds or substances that are cancer causing, so carcinogens. Aniline dyes, cigarette smoke, asbestos, these things are known to have a link to cancer. So being around these things can give you cancer. You want to take particular care when you're around these things. There are viruses as well that can cause cancer. RNA viruses, DNA viruses, and again, these some of these are genetic, meaning I guess that's not the right word. They're all genetic. Some of these are like family, like they occur within families. So there may be like a missing or a defective gene as opposed to a mutation that's caused by one of these. To give you an example, I was working with a bacteria that I had a bacteria, and I had a medium called 2,4-D. I forget the rest of the name. It was it's not a pesticide. It's used in grass to make grass greener. Anyways, I wanted to make my bacteria be able to eat it and use it as a food source when they couldn't originally. So I had a bunch of bacteria on this plate that had 2,4-D in it, and of course they were all dead. None of them were growing. And what we would do is we would just shine UV light on them for like 10 seconds, and then like come back the next day, and then shine UV light on them for 10 seconds. And bacteria are so small, and their replication, and they don't have much protection from things like UV like we do with our skin and stuff. But they replicate so fast that it's easy to see when mutations happen. And so just by exposing them to some UV light repeatedly, we caused their DNA to have so many mutations in it. So one of the things that happens with UV light is it causes thymines that are next to each other to form dimers. And this can often cause DNA polymerase to kind of get stuck when it gets to that point and not know what to pair with the two T's, because instead of the two T's having complementary base pairing, they're like stuck to each other, right next to each other, and so the DNA doesn't know what to stick in there, and so it just sticks something in rather than not stick something in, right? If DNA polymerase was like, I don't know what to do, and it just stopped doing things, people would die. So it just goes, okay, well, here's a G, here's an N, whatever. Not an N, but and so UV light can cause, deletions or insertions or point mutations, and those can very quickly add up and lead to, um, you know, um, diseases or cancers or misfolded proteins. But having a missing gene altogether, or having, just defective genes and passing those down from generation to generation is also a possibility. All right, one more learning check. So here, try to match these up as best you can. Pause it, give it a shot, and then here is the end of the chapter. So this one had a lot in it. It was kind of long. We talked about the nucleic acids first. We transitioned into transcription and translation. Don't get bogged down with a lot of the details. We didn't go over very many details. I mentioned a few enzymes that were important in replication, a few enzymes that were important in transcription and translation. We talked about regulation a little bit with transcription factors, but don't like worry about that too much. Know what their job is, but don't worry about like all the different. If you like Google something online, you can get bogged down with just the immensity of the information on transcription factors alone. So just kind of focus on the basics. Know the types of mutations and a little bit about what we talked about viruses. But if you have questions, of course, email me like you guys usually do, and I will see you in the next one. All right, guys, take care.